Uh, Good afternoon, everybody. We're going to get started. I'd like to welcome you all to this seminar today. This is a little unusual in that we're co-hosting a seminar. This is sponsored by the Levy CIMAR. My name is Brian Noonan. I'm Executive Director of Levy CIMAR. We're co-sponsoring and co-hosting Dr. Cowman with the Tufts Center for Global Public Health and the Tufts Global Health Faculty Council. I'm going to invite Dr. Honorine Ward to come over and introduce Dr. Cowman for us. Thank you.
Thank you, Brian, and welcome everybody. I'm really delighted to introduce our speaker today. Professor Alan Cowman is Acting Director of the Walter and Eliza Hall Institute, a very prestigious research institute in Australia, which you can see here. Professor Cowman is internationally recognized for his expertise in malaria research, which he has had a distinguished career for over 30 years. He obtained his PhD at the University of Melbourne and the Walter and Eliza Hall Institute. He is a fellow of many prestigious academies and societies, including the Royal Society of the UK, and he has received numerous awards and honors. Professor Cowman has over 360 peer-reviewed publications and serves on several editorial boards. His lab focuses on research aimed at understanding how Plasmodium falciparum invades and survives in red blood cells with the goal of developing novel antimalarial drugs. Today, he will be speaking to us about discovery and development of novel antimalarials and their use as chemical tools to discover the new biology of the malaria parasite. Professor Cowman, thank you so much for visiting Tufts and coming to speak to us today.
Thank you very much, Honorine, for that generous introduction. Really great to be here at Tufts University. First time I've been here, although I've been to Boston a number of different times. So thank you very much for allowing me to talk to you and tell you about some of our data that we've been generating. I'd like to talk about a project that's been going for about 6 or 7 years in terms of development of new antimalarial drugs. And this started with a phone call from Merck in the USA, David Olson, who works out of the West Point Merck organization just out of Philadelphia, rang me one day and said, we've been following your work and we believe you've got some really good targets for potential new antimalarial drugs. Would you be interested in collaborating? And it took me a few milliseconds to say, yes, we'd love to collaborate. And then he said, but, and then the but was that Merck couldn't give us any money and that to support any work that we were doing and they were doing, we needed to raise money. And so we then joined together and applied to the Wellcome Trust for a number of different grants, and Wellcome Trust has been funding this work for the last 6 or 7 years reasonably generously. So what I'd like to, I'll just give a quick introduction on malaria because I'm sure quite a lot of you don't actually work on malaria, and I'm sure you do know that it's a big problem with respect to the number of people that get infected and that die from malaria each year, with about 600,000 deaths now each year. There is a vaccine, RTSS, which is about to be implemented, and it's not particularly good. Only 39% protection in children after 4 doses. And so there needs to be a lot of improvement, but it will save lives, and that's why it's being implemented. So over the last 20 years, there's been a really good news story with respect to malaria, where the number of malaria deaths has decreased by about 50%. And that's really an amazing story that has resulted from increased spending on control of malaria, deployment of bed nets, and other interventions. So calculated to have saved more than 4 million people from death over that time period. So really a good news story. However, there is signs of malaria resurgence, and COVID hasn't helped because there's less money being spent on control of malaria. So you're seeing resurgence of malaria in different places around the world. There's also an issue with respect to resistance of mosquitoes to pyrethroid insecticides, which are used to impregnate bed nets. And also the main treatment and control of malaria is artemisinin-based combinations, or ACT therapies. And there is decreasing efficacy of these combinations, and really resistance is starting to develop and spread around the world. So we do need new classes of antimalarial drugs. We need a pipeline to take the place of drugs as the parasite develops resistance. And it's one of the stories that I'd like to talk to you about. And really this started quite a long time ago, because we were analyzing the role of proteins in export of proteins into the red blood cell, because the malaria parasite infects the red blood cell and goes through a cycle from what's called ring stage to trophozoite stage to schizont stage. The merozoites then burst out and then reinvade. And you see that part of that cycle there up on the screen on the left. We'd identified an important aspartic protease that is required for export of proteins out into the red blood cell. So that aspartic protease identified proteins for export, cleaved the N-terminus and in the endoplasmic reticulum, and then put them on the pathway towards export. And so that was the target that Merck and we were particularly interested in as a potential drug target. So we wanted to do a high throughput screen with that target, and we've done that. Turns out that the SAR of that particular target is very flat and very difficult to develop small molecules, although peptidomimetics work really well in terms of potency. At the same time, we were doing a phenotypic screen where we had obtained a library of compounds from Merck that they developed in their different programs, such as high blood pressure. And they sent us a representative subset of those compounds, about 1,300 compounds. And we did a P. falciparum phenotypic screen in vitro to identify those that would kill the parasite. And that identified 32 hits, and they all belong to the same drug-like hit class. And you see the structure of one of them there at the bottom. And that one we have called WM4 for WEHI Merck 4. This is an example of the potency of these hits. And this is determining the EC50 of P. falciparum viability, and I'm comparing it to chloroquine. And what you see is that the EC50 for chloroquine is 15 nanomole, and chloroquine is one of the wonder drugs of the 20th century. WM4, one of our initial hits, is more potent than chloroquine in vitro, which was a huge surprise. We were expecting hits that might be in the micromolar range, or at least in the very high nanomolar range. So to get something that's really that potent was really quite amazing. And even more interesting was that it had in vivo activity at the same time, even though it wasn't particularly a drug in terms of its properties. So in terms of, as we didn't know the target at that stage, we needed to then work out what the target was of those drugs. And the way that we normally do it in Plasmodium falciparum is to select resistance, which is a bit counterintuitive, because what you want to really do is select drugs that have a very high barrier for selection of resistance. But I'll come back to that in a minute. As you can see that we were able to select resistance. It wasn't easy. It took about 6 months of slowly increasing the level of drug until we got around 14 to 16-fold resistance to WM4. And there's a second compound there, WM5. We then sequenced the genome of those resistant lines and found only one change in those resistant lines, and that was an amplification event on chromosome 8. P. falciparum has 14 chromosomes. Within that amplification event, and we had a number of different amplification events in different selections that we did, and they were different sizes. This one was the smallest one. And the most obvious and important gene that we identified was plasmepsin 10, which you see there with the red arrow. And we obviously thought, okay, that's the target, or at least the mechanism of resistance, because nothing within that amplicon looked particularly interesting in terms of the mechanism of action of these potential drugs. And you can show that plasmepsin 10 gene is amplified and overexpressed in those different lines. Okay, so we had a starting point, a really good starting point in terms of hit to lead development for these compounds, starting with WM4. And then we set up this collaboration with Merck, but also WEHI Aptec in Shanghai, who synthesized the compounds. Compounds were designed with Merck and WEHI. We then developed a number of different assays where we could screen the compounds, including structural biology. So we, from the identified target, solved the structure of plasmepsin 10 with drug bound in the active site. Design new compounds around that, and then had them chemically synthesized at WEHI, who then shipped them back to us. And so we went around that cycle around 5,000 times. So there have been 5,000 compounds that have been designed and synthesized in this program. We then ended up with this compound, WM382, which is what I'll call the lead or tool compound. And so most of the biology that we've been doing has been with this tool compound, WM382. During the development of WM382, we realized that there were a subset of compounds that were much more potent. And we already had really high levels of potency in the low nanomolar range, but we had a subgroup that were in the low picomolar range in terms of its EC50, so really potent. So we thought the only way we could get that potency is if it's hitting more than one target. And the most likely target that we thought that we could potentially hit was plasmepsin 9, which is closely related. And this shows the 10 plasmepsins encoded by Plasmodium falciparum. 1 to 4 are present in the food vacuole, and they're responsible for digestion of globin in the food vacuole, and they're not essential, so we didn't think they would be particularly important. Whereas plasmepsin 9 and plasmepsin 10 are essential. We and other groups have shown that by doing knockouts, conditional knockouts. So the second target is plasmepsin 9. I'll show you a little bit of data that shows that. And we proved that in a number of different ways, but I'll show you actually some very recent data that proves it very nicely, and that's using thermal proteome profiling and proteomics, where essentially we use thermal PISA with WM4 and also WM382. And what you see here with respect to that assay is a signal of one protein where it's being stabilized during heating by binding of WM4, and that's plasmepsin 10. What that tells us is the target for WM4 is plasmepsin 10. If you do the same thing with WM382, you again get plasmepsin 10 as you would expect, but you also get plasmepsin 9. So it proves quite nicely that WM382 is binding to both plasmepsin 9 and 10. Interestingly, we got a third signal that is significant, and that was plasmepsin 1. As I said, plasmepsin 1 is in the food vacuole. It's involved in digestion of globin. You can knock it out. It's not essential. We don't think it's important in terms of killing of the parasite. Okay, so just to show you the activity of all the compounds that have been developed in this program, and this is plotting plasmepsin 10 activity or the ability of the different compounds to inhibit plasmepsin 10 activity, and plasmepsin 9 activity along here. And as you can see, it's really easy to design compounds that don't inhibit plasmepsin or 9 or 10 at all. And that's actually most of the compounds that we designed. So rather than getting more potency, we actually went the other way, as you might expect. WM4, really selective for plasmepsin 10 over plasmepsin 9, as I've already told you, and mixed in amongst a lot of different compounds. So relatively easy to make plasmepsin 10 specific compounds. Quite hard to make compounds that inhibit both plasmepsin 9 and plasmepsin 10. WM382 is the dual inhibitor. This one, MK7602, is actually our clinical candidate, which I'll talk a bit more about. In terms of plasmepsin 9, we've been trying very hard to make selective compounds for plasmepsin 9, not as drugs, because we really wanted to make sure we dialed in the dual specificity against plasmepsin 9 and 10, because if you're hitting 2 targets, you're going to get a much higher barrier for resistance, which we do. Plasmodium 9, we would love one that really hit that, but we've tried a bit, but without success. So for some reason, we can't make a Plasmodium 9 selective compound. In terms of the structure-activity relationship, I won't go into this in any detail because I'm not a medicinal chemist. But what you can see here are the two things that I think are important. This here with respect to the stereochemistry, and very important that we had an R stereoisomer here. The L stereoisomer essentially has no activity. And second here in terms of the stereochemistry where we need S enantiomer. So a lot of work went into this in terms of understanding the SAR around this core part of the molecule to develop compounds that were essentially drugs and good antimalarials. That included structural biology. And we have a number of different structures with different compounds in the active site. This is just one. This is plasmodium 10 with WM3A2 bound in the active site. And in red, the red patches are the aspartic acid amino acids that are absolutely critical for the activity of this aspartic protease. And using structural biology, we were able to do a number of things. We were able to understand why WM4 is a specific inhibitor of plasmodium 10 compared to plasmodium 9. We were able to understand how it interacts with the active site and the important interactions with respect to that. We don't have a plasmodium 9 structure at this stage, but we used homology modeling. Interestingly, that homology modeling with plasmodium 10, which we started doing before we got these structures, actually was not quite correct. We had the wrong shape of the compound, and this enabled us to correct that and help considerably in terms of design of new compounds. In terms of binding of WM3A2 and the clinical candidate MK7602, we were able to show the different interactions. And an important aspect of this is that we get induced fit when these compounds bind. There was a flap that we couldn't resolve without the drug binding. And once the drug bound, it essentially bound to that flap, and its mobility stopped, and we could actually resolve that. So there's an induced fit and increased interactions of this compound with different parts of the active site. And that explains why we have such high affinity, which I'll talk a little bit about in terms of how these compounds bind to the active site. So this provided key information for structure-guided design of inhibitors. So we then went on to develop an optimized lead and then a clinical candidate, which is now called MK7602. And that's the structure of that compound above it. This compound is now in clinical trials. It is in phase 1 clinical trials, and safety profiles are looking extremely good. Just a bit of enzyme kinetics here, just to show how well these compounds inhibit both plasmodium 9 and plasmodium 10. And this is the EC50, the ability of these compounds to inhibit the parasite in vitro. And you see they're very potent, 0.42 nanomolar, 0.4 nanomolar for these two compounds. This is their ability to inhibit activity of plasmodium 10 and plasmodium 9 over here. This is Plasmodium falciparum, this is Plasmodium bivax, plasmodium 10. And you can see very much in the low nanomolar, picomolar range. So very potent in terms of inhibiting both falciparum and bivax, plasmodium 10, and also plasmodium 9. So they are both dual inhibitors of both of these aspartic proteases. And the fold selectivity, we wanted to keep this as low as possible. And so in moving from WM3A2 to MK7602, that fold selectivity of plasmodium 10 over plasmodium 9 dropped down to 5-fold. So obviously we want these antimalarial drugs to hit P. falciparum, but also P. vivax. P. falciparum is the most deadly in terms of malaria parasites, but P. vivax is very important as well. So we collaborated with Jean Popovich at the Institute Pasteur in Cambodia. These are difficult assays because what they're doing is waiting for people to walk in infected with P. vivax and then taking blood and doing ex vivo experiments to determine whether the compounds inhibit the growth of the parasite, because you can't grow P. vivax in vitro, at least for any length of time. Again, the EC50 here for these compounds. These are both dual inhibitors, and this is the plasminogen 10 selective. And these are the number of P. vivax isolates that have been tested. You can't get exact numbers with this assay. But in terms of killing P. vivax, the EC50 is less than 1 nanomolar for both 7602 and 382. And for this plasminogen 10 selective, it's quite a bit higher, but somewhere between 1 to 10 nanomolar. So that was good news. We've proved that it kills P. vivax. We know that it kills P. falciparum, at least in vitro. So we had in vitro potency. And this is just the figures in terms of potency on the parasite and the proteases as well. So what about safety in humans? And unfortunately, I can't really talk about the PK/PD data. It will be presented at the ASTMH conference in Chicago in a couple of weeks. But so that'll be the first place that we're able to reveal some of the information. But I can say that the preliminary human dose prediction is around 24 milligrams. The PK and PD looks extremely good. In terms of the safety profile is also looking good in terms of cardiac properties, which is great, and also in terms of turnover in the liver. So as I said, MK7602 is currently in first-in-human clinical trials. This is the synthesis of MK7602. I'm not a medicinal chemist. I'm not going to go through this in any detail. But as you can see, it's a number of steps, so it's a little complex at the moment. However, Merck have an amazing team that whose role is to get the number of steps down and to make the synthesis of any potential drug more cheaply. And for malaria, that's really important. It's really important to have a really low cost of goods. An interesting point is that this enables convergent synthesis from commercially available building blocks. So there is a really good chance that the cost of this compound would be really quite low. Just a little more on the ability of these compounds to bind their target. This is surface plasmon resonance using Biocore. And what you see here is WM4, remember, is the plasminogen 10 specific compound, using SPR to try and work out the affinity of binding, so the KD. And as you can see, there's a really fast on rate and then a slow off rate with a KD of about 0.48 nanomolar. So quite tight binding. When you look at plasminogen 9, and remember this compound is selective for 10, very fast on rate, very fast off rate with a KD of about 77 nanomolar. So you can understand why this is plasminogen 10 specific and not particularly good at inhibiting plasminogen 9. So let's compare it to WM382 and MK7602. P. vivax plasminogen 10, a fast on rate. The off rate is not measurable. You essentially can't see an off rate. So these compounds bind extremely tightly, and you can't actually measure an off rate. And so the best we can do in terms of a KD is that it's less than 0.1 nanomolar. If you look at plasminogen 9, you get exactly the same result. A very fast on rate, and then an off rate that you can't really measure, and a KD of less than 0.1 nanomolar. So these bind very tightly. They have a very slow off rate, and that's probably really important in terms of the high potency that we're seeing for these compounds. How do they kill the parasite? And essentially, we've done that by growing the parasite in the presence of blood over a full asexual blood life cycle, and you can show that they essentially stop the growth of the parasite towards the late schizont stage, but they also stop invasion of merozoites. If you break them out of the schizont and ask, can they invade? They can't. So they're not able to egress. They're not. If you do break them out, they're not able to invade. And this is just an egress assay showing the ability of both WM4 and WM382 to block egress. And on this movie, which I think will work, I'm not quite sure how to do it. No, won't work. Okay. Very nice movie. What it showed on the left there was wild-type parasites untreated with drug, and all of them are infected with malaria, falciparum parasites, and you would have seen the merozoites breaking out and moving over the field. On the right are those treated with WM382 in this example, which are essentially sitting there moribund and apparently dead. What about their ability to cure mice of Plasmodium burgii infection, which is the mouse model of infection that we use quite a lot? And the answer is that they can indeed cure mice. And this is just one example of an experiment where we've given infected mice with Plasmodium burgii and then given them 4 doses of drug QD once a day, and then asked, does that cure them of malaria? They become parasitemic quite early at about day 8. If you treat them with chloroquine, 10 mg per kilogram, they become parasitemic at about day 18. Whereas WM382 at 60 and 30 milligrams per kilogram cure the mice of infection. And this is just one experiment. You can go down to 1 milligram per kilogram in these experiments and it cures the mice. As much as we'd love to cure the mouse world of malaria, we're more interested in humans. And so what we turn to is a humanized mouse model where these have been essentially, the mouse red blood cells have been replaced by human red blood cells so that we can infect them with P. falciparum in the blood. And so these mice have been infected at day 0 with P. falciparum. And then that parasitemia has been allowed to increase up to about 1%. Then they've been treated 4 days in a row, QD, once a day, and then parasitemia followed over that time. And so you see parasitemia increasing without treatment. With chloroquine, you see suppression of parasitemia. With 3, 10, and 30 milligrams per kilogram, you see the same level of suppression as chloroquine. And at 1 milligram per kilogram, you see partial suppression. That's good news. WM382 is at least as good as chloroquine in terms of its ability to cure mice of both P. burgii and P. falciparum infection. In developing any new antimalarials, we want them to hit the blood stage, but we also want them to hit other stages of the malaria parasite. And this is the life cycle here. This is what I've been talking about here, the blood stage. We would like them to also hit the liver stage. And so these experiments are determined to test that, where we've infected mice with Plasmodium burgii in this case, and either 5 or 40,000 sporozoites or 5 mosquito bites with mosquitoes infected with Plasmodium burgii. And then treatment about 36 to 48 hours later with different doses and either one or a number of different doses. And these P. burgii express luciferase, and we can detect the presence of parasites by injecting luciferin. And you see the liver here is highly infected with no treatment, as it is with WM382 in both of these cases. You see the liver infection then decrease over time for no treatment and then spreading around the blood of the mouse. However, you do not see that with treatment with WM382. What you see is essentially nothing beyond the liver stage. And what we've determined is that these parasites are actually able to egress from the hepatocytes that they've infected here, but the liver merozoites that are being released here in this part of the life cycle are not viable, and they can't invade red blood cells. So we end up being able to cure at liver stage at different levels of drug treatment with respect to WM382 and also with MK7602. And this is oral delivery. Yep. What about transmission? And so we wanted to test the ability of these compounds to block transmission to the mosquito. And this is P. falciparum in this case, not P. burgii, where we're growing them in vitro and then pushing towards development of sexual stages that the mosquito takes up, but treating those sexual stages with either MK7602 or WM382, allowing the mosquitoes to feed on the blood and then allowing any parasites to develop in the mosquito, and we dissect out the midgut. And this is a midgut from a mosquito that's infected with P. falciparum. So what you're doing is counting these round dots here, which are the oocysts that have developed within the infected mosquito. If you treat them with WM382, in this case, you see no infection of the dissected mosquito gut. And you can quantitate this, and as you see here, no treatment. You see more than 20 oocysts per mosquito gut. Whereas if you treat with drug at 30 nanomolar, you see absolutely no oocysts. So these compounds are able to block transmission to the mosquito, which is, again, exactly what we want. So what does that mean in terms of clinical use of these compounds? Firstly, as I said, they block liver stage, they block blood stage, which is here, and they also block transmission to the mosquito. So they can certainly treat infection, but they would also block transmission. So they're excellent in terms of treatment and prophylaxis, and hopefully for elimination of programs of malaria. So if we do develop antimalarial drugs, we want them not to show cross-resistance to already existing antimalarials, but also to antimalarial drugs that are being developed now. So they're in the pipeline, where we know that there is resistance already and that the mechanism is known. And so what this is doing is these are parasite lines that are resistant to different drugs, including this 3D7 WM4 line, which we selected, that is resistant to WM4, that has plasmodium 10 amplification and overexpression. I've taken out the numbers here and just coded them either red or green. Green means no cross-resistance, red means cross-resistance or resistance. And you see that most of these are resistant, as we expect, to the different compounds that include tovaquone, chloroquine, mefloquine. This other one here, KAE609, which is currently in the pipeline of clinical trials. WM4, resistant, as you'd expect, and also this plasmodium 10 specific compound that also hits plasmodium 10 shows cross-resistance as well. WM382, a dual inhibitor, does not show cross-resistance. So plasmodium 9 alone is sufficient to get over the resistance that's been developed with amplification of plasmodium 10. We see no cross-resistance with any of these lines. So it's possible to do this in a more elegant way, and Marcus Lee at Wellcome Sanger Institute has developed this, where they've made around 50 parasite lines where they've inserted the mutations that confer resistance, the known mutations, and then labeled them or barcoded them at the same time. Then you can mix all of these 50 parasites together and then put your drug on and select it. And if parasites go out, you can sequence it and then work out which particular resistant mechanism might be involved in whatever your drug is. And so we sent a number of different compounds to Sanger Institute and asked them to do that with this RBAR platform that they've developed. And these are the compounds that we sent them here, included MK7602, WM382, and also WM4, and a number of other compounds. This is our control with no drug. This is our positive control of a parasite line that has mutations for this particular drug. And these are all the ones that we sent to Merck. And as you can see, nothing grows out. So there is no cross-resistance to these compounds, which is good news. What about resistance? Can we select resistance to these compounds? And I'll just show you one way of testing that, where we take P. falciparum parasites and we challenge them with larger concentrations of drug, and then ask, can they recrudesce and grow after that insult of drug treatment? And these are the results here. Firstly, a tovaquone, relatively easy to select resistance. And as you can see here, it takes between 30 and 60 days for those parasites to recrudesce. This is our, again, a positive control, CAF156, that is again in the pipeline of development for new antimalarial drugs. And as you can see, it's even easier than a tovaquone to select resistance, coming out at about day 18 to 30. WM4, which is plasmodium 10 selective, we've never been able to select resistance using this method. So it's still very hard to select resistance, even with a single target compound of this class. WM382, and this is MK7602, what you see is that we have not been able to select resistance to either of those compounds. So the dual inhibitors, it's very difficult to select resistance. They have a very high bar for the development of resistance. Not unsurprising since they're hitting two targets. And so you need changes in both of those targets at the same time in order to for the parasite to be resistant. So you can do a risk assessment of resistance, and this has been done here in a spider chart for atovaquone. Essentially what this is doing is looking at the minimum inoculum for resistance, and that is quite low for atovaquone, and so it's plotted here, and you end up with this spider chart. So atovaquone is in blue. You can also measure the shift in IC50 or EC50 for the parasite, whether there's an in vitro resistance genotype, the time for recrudescence, whether there's an in vivo resistance genotype, and whether there's pre-existing resistance in the field. And so the answer is yes in the case of all these, and you can actually quantitate that and make this plot. What if you do that for WM382 and MK7602? You can't do it for all of these because we don't actually have them. There is no pre-existing resistance in the field. There is no in vivo resistance genotype, but the best we can do is essentially plot that, and what they end up with is a single dot in the middle here that tells you that there is a very low risk for resistance for these compounds. What is the biological function of plasmapsin 9 and 10? And obviously, in developing these compounds, they're really useful tools for understanding the role of plasmapsin 9 and 10 in malaria parasites. And so what we did was use bioinformatics to try and find proteins that we thought might be processed. And we developed an assay, a FRET-based assay that enabled us to assay activity so that we could then design peptides that had those sequences that we thought might be cleaved by these different proteases. And these are some of the experiments here where from different proteins in P. falciparum that we thought might be cleaved, in particularly by plasmapsin 10, but also plasmapsin 9. We then came up with a list of potential targets and also a motif that appeared to be the cleavage site for both plasmapsin 9 and 10. And actually, the motif for 9 and 10 is virtually identical. Both of these proteases seem to be able to cleave very similar sequences, and they cleave after the phenylalanine sequence here on this motif. What we can show that plasmapsin 9 and 10 are master regulators of P. falciparum egress, where merozoites are trying to leave the red blood cell, but they're also, sorry about that, they're also regulators of invasion into human erythrocytes. And it's interesting how the research you're doing sometimes ends up coming like this, where you're moving along two parallel streams, and all of a sudden you find out that these compounds block invasion, and you've been working on invasion for the last 30 years. And so you have all of the antibodies and the tools to really analyze it really quite quickly. And this is at least some of our understanding of the invasion process. And what I'd like to talk a little bit about are these proteins here, the erythrocyte binding antigens, which bind receptors on the red blood cell, and the PFRH proteins, which also bind specific receptors, as well as this complex here that binds Basigen on red blood cells. We've been trying to work out, we knew the processing of these ligands was very complex. We were trying to identify the protease for a couple of years. Once we had these compounds and we knew the motif, we looked at these proteins and lo and behold, where we knew they were processed was a nice cleavage site for plasmapsin 10. And this is just a few examples of how we defined the substrates of these different proteases. This is the apical sushi protein. It is, as we know, plasmapsin now is plasmapsin 9 processed. And having these compounds, you can essentially treat parasites with either WM4. We know it's plasmapsin 10 specific. WM382 is a dual 9-10 inhibitor. This protein is not inhibited in terms of its processing by WM4, but is by WM382. So we know it's plasmapsin 10. We can then purify the protein and by mass spec actually show the cleavage site, which we did. You can do the same thing for another example. This is a PFRH5, and you can show that it is processed from 65 kilodaltons down to 50 kilodaltons. WM4 inhibits it, as does WM382, and we know that plasmapsin 10 actually processes this protein. And so from that, we and others, and so there was two really nice papers in Science from Dominic Soldati's lab and also Dan Goldberg's lab, which identified a number of these substrates. We were able to enlarge that quite significantly, and all of these proteins in here, the RHs and EBAs, AMA1, this protein here, RIPA, PTRAMP, are all processed by plasmapsin 10. We're also able to increase the number that are within this plasmapsin 9 substrate list. We also showed that both of these proteases are autocatalytic. They activate themselves. They also activate other proteases such as subtilisin 1 and subtilisin 2. So if you block their function, you essentially block processing of really important invasion ligands, important proteins involved in egress, as well as other functions. Okay. Hard to see that from my angle. So the next thing we wanted to do was understand, and I'll just show one example. We wanted to understand what the function of some of these processing events were. And so we chose two in particular. We chose this complex here that we knew most of the proteins within this complex are processed by plasmapsin 10. So we started by mutating RIPA, which is cleaved in the middle. So from around 120 to a 60 kilodalton, 60 kilodalton fragments. And we mutated that cleavage site that we'd already shown by mass spectrometry to ask whether it was essential. And the answer is it isn't. The parasite grows fine without that cleavage site. And that actually didn't surprise us at all. But the next one is important. And I'll only show one result because this involved a lot of different mutation, because it's not as simple as I'm going to tell you. It's quite a bit more complex. And that's PFRH5, which is processed by plasmapsin 10. And so we started mutating that cleavage site, and mutations are essentially not viable. And so what we did was to express a mutant form on top of a wild type form that we had under conditional control. So we can turn this one off and leave this one on and ask whether the parasites are still able to grow and invade. And just a long story short, this is the most important mutant where we leave off the N-terminal section. And so it's essentially just like an already processed product. And we can show quite clearly without that processing event, the parasites are not viable. So that processing event is absolutely essential. What's it doing? PFRH5, as you remember, binds pathogen, absolutely critical for the invasion process, as is the other members of this complex. So despite the fact that this is a long story, in the end, it turned out to be quite simple. And probably this is the last possibility that we tested, and probably should have started first, and that was whether the unprocessed form actually bound pathogen versus the processed form. And the answer is the unprocessed form cannot bind pathogen. It has to be processed so that this complex can bind pathogen. And you can show this quite clearly here with the processed recombinant form versus the unprocessed recombinant form. Unprocessed, full length, cannot bind red blood cells and cannot bind pathogen. The complex with RH5, this complex altogether, this is recombinant. With RH5 processed, can bind, unprocessed, cannot bind. So you require processing of RH5 by plasmapsin 10 so that it can then bind pathogen for the invasion process. If you have a full-length RH5 without processing, cannot bind pathogen. What's the reason for this? We're not sure at this stage, but we think there's probably something deleterious about having this form which can bind pathogen quite early. And so it's really quite finely timed in terms of when plasmapsin 10 cleaves PFRH5 so that it's active. So plasmepsin 10 actually activates the function of this complex. All right, just looking at the time. Okay. So just very quickly, I'll just tell you a little story that, again, is a bit longer than what I'm going to tell you, but we were interested in understanding if you actually, and this comes from the story of chloroquine being used to block red blood cell in development, and asking if you do that and then challenge mice or humans down the track, are they immune? And when you do that with chloroquine, they are actually immune. We wanted to test this in mice with respect to these compounds. And we wanted to test it at a specific blocking point, and that's in the liver. If you block their development in the liver, so that you don't get viable liver merozoites, does the mouse develop immunity? And this is Plasmodium burgii. So we've gone back to the mouse malaria, where we've infected mice with 40,000 sporozoites. Sorry, I can't see it. 40,000 sporozoites on the left there, and then challenge them with P. burgii by infecting them with mosquito later on. And what you see here is naive mice where they've been infected, and you see liver infection here. If they've been treated with WM382, they don't go beyond that, and so you don't get blood stage infection. If you test them for immunity 3 months later, they're immune, and that's comparing naive to mice infected with P. burgii that have been treated twice with 100 milligrams per kilogram of WM382. And you see very strong immune responses and immunity at 3 months, at 6 months, and at 19 months. So with a single immunization with WM382 and parasites that block at the liver stage, you get protection against mosquito reinfection for up to 19 months. We know that immunity is CD8 T cell, or CD8 positive T cells are responsible for it. And these are the experiments that show that, that here, where we've infected mice with 40,000 sporozoites, cured them with WM382 at the liver stage. 8 weeks later, we've essentially boosted them again with 20,000 sporozoites and then cured them at the liver stage. And then 10 weeks later, tested them for their immunity, but depleted the CD8 positive T cells. And what you see here is the experiment from CD8 T cell depleted mice versus naive mice versus those that have been treated with, that still have CD8, a full CD8 positive T cell complement. And you see immunity here and recrudescence at this stage. So we know that CD8 positive T cells are necessary for sterile protection from WM382 vaccination combination. And we can show that in particular liver resident memory T cells, if you deplete them, that they're responsible for that, particularly important for that immune response. And this is an isotope control versus where we've depleted liver resident memory T cells. So what's happening? Essentially, what we're doing is blocking the development of P. burgii at this stage. We're still getting release of liver merozoites, but these are not viable. We're getting an immune response to these that is having an important effect on immunity at both the sporozoite stage and at the hepatocyte infected stage. At the hepatocyte infected stage, CD8 positive liver resident T cells are really important. We're also getting antibodies that are blocking, helping to block sporozoite invasion of hepatocytes. So I've just given you the short version of the story, which is actually really an added benefit of these drugs, that not only do you stop infection, but you also may well get increased immunity with respect to them. So what I've talked about is identification of novel class of compounds that are potent inhibitors of malaria parasites. I've shown that oral dosing of these compounds can cure mouse models of malaria, including P. falciparum. I've shown that WM382 and MK7602 don't show cross resistance to the current antimalarials as well as those that are being developed as that also that there's a high bar for selection of drug resistance for these compounds. I've also shown that they block at the liver, at the blood stage, and also at the transmission stage to mosquitoes. I've also shown that they have target product profiles that are useful for treating, will be potentially useful for treating patients, but also for chemoprotection. MK7602 is now in first-in-human clinical trials, which is going extremely well. And the dose predictions are looking very good. WM382 and MK7602 will induce sterile hepatic immunity, and I just gave you a little snippet of that story. And we've used those chemical tools to understand the function of plasmodium 9 and 10 aspartic proteases, and to show that they are master regulators of a number of steps in the development of the parasite. And also, we've used those compounds to understand the structural basis of inhibition, as well as the role of particularly plasmodium 10 in invasion in the example that I showed you in processing of PFRH5. These sorts of projects aren't done by a few people, they're done by large teams. We have a large team at WEHI who's been involved in this project, and names are there, and I won't go into them in detail, except I'll mention Paula Favutsa, who's the project manager. Working with Merck over the last 6 or 7 years has been absolutely fantastic. They are great collaborators. And the amazing expertise they've got across the area of drug development is just incredible to see. I've learned so much about drug development over the last 6 years. Really enjoyed it. And I'd like to mention David Olson, who's the leader of the teams at Merck, as well as John McCauley, who's the head chemist, and Manuel Ruiz, who is one of the main chemists involved in designing the compounds. Really an absolutely fantastic collaboration between WEHI and Merck. But I'd also like to mention WEHI Aptec, our CROs, who synthesized the compounds that were designed, and also the funding from Wellcome Trust, which was absolutely critical to this project. Thank you very much.
Very much for that amazing talk.
Yeah, so these compounds are very selective for 9 and 10, with perhaps a little bit of activity on plasmodium 1 as well. So they don't hit the SVP at all. They obviously don't hit the human aspartic proteases either, really important, and the safety profiles are looking really good with respect to that. Would there be good starting points to try and develop drugs that hit SPP? Maybe, and vice versa. Certainly, these drugs might be interesting in Cryptosporidium and perhaps other parasites, and obviously that's an area of interest. And they may well be hitting aspartic proteases in those other parasites. And in any case, they would be good starting points to do that, absolutely. So I think there's quite a few possibilities in terms of using them as they are, or potentially starting points for development of drugs against other proteases and even other parasites. Yeah, agree. Yeah, certainly with conditional knockouts to SPVP, they're dead. Yeah, so it's a very important protease. Yeah, there's a lot of different targets of those. So just mutating one probably would be lethal. So you have to have a conditional. But so I'm not quite sure what you're getting at. Yep. Yeah. Yep. Yeah. Yeah, I think the last part is the important question. What are the targets for the immune response? And the answer is we don't know. For the antibody response, CSP is one of them, but it's not the only one. And it's not the most important in terms of blocking sporozoite invasion of hepatocytes. For the T cell response in for falciparum infected hepatocytes, we're not sure what the antigen is. Antigens probably. And that is a whole new project in terms of identifying those antigens. They're probably really important to do. But I think what those results show is that you're getting that side benefit of not only killing the parasite, but getting immunity, potential immunity as well, which I think is interesting. Yeah. Hard to do that with Bergii since there's really only one good strain. So it hasn't been done. So obviously needs to be done and could be done, I think, with Yoelio, but that hasn't been done yet. Probably may do that as a human challenge model. And I think that would be interesting to look at. So at this stage, we don't know whether we're getting protection against different strains. Yeah, so I just showed you one example of one experiment, and those haven't repeated in multiple experiments, whereas with plasmodium 10 and 9 and plasmodium 1, that has repeated. So that was just an example of one experiment. So those other ones that look down-regulated are not relevant to the experiment. So it's not reproducible. That's correct. Yeah, but plasmodium 1 is, which was very unexpected. Yeah, it's not covalent. It's just binding very, very tightly, and the off rate is extremely slow. So we can't actually measure it. We haven't worked that out at this stage. It's it'd be worth doing that, but we just haven't done it. Thank you. Hello. Oh, you're welcome.